Hello. Hello, everyone. Good afternoon. Thank you for coming. I'm very glad to see so many people interested by the topic of today. So, as you have heard yesterday, we have a change in the program. As Pegginopoulos couldn't make it, however, as you will see, I will introduce the Radhia Kacher when it's our time to talk. We will have a great session talking about brain development and juvenile pediatric HD, Huntington disease. We all are aware that the mutant Huntington gene is expressed since the beginning and that brain development is altered in Huntington disease. However, there is still many unanswered questions, among them whether the altered brain development contributes indeed to the pathology, to the adult pathology. What are the causes of the variability and in the disease expression and progression? Does pediatric form of Huntington disease correspond to the developmental form of Huntington disease? And in a more general question also is what are the pathological and compensatory mechanisms engaged from brain development to avert the overt phenotype in adulthood. With that in mind, I will introduce the first speaker, that is Mariacristina Capizzi. She's an Italian but working in France, so maybe we can have her. So she will tell her about the consequences of abnormal axonal growth on early Huntington disease signs. So may I have the next slides? We can go to our presentation. No, okay.
So, good afternoon to everybody. I would like to thank the organizer to invite me to and give me the chance to speak about axonal growth in Huntington disease mouse model. My presentation will have four main axes of discussion, and I will start introducing you the developmental hypothesis of Huntington disease and why we get interested in the axonal growth. Then I would like to finish to discuss with you, to open a discussion about the possibility to candidate the microtubule cytoskeleton like a new therapeutical target. So Huntington disease is an adult disorder and is caused by the mutation in a Huntington gene. The protein of Huntington is expressed since the beginning of development. This means that the HD fetuses are exposed to the toxic function of the protein since the beginning of their formation. Recently, has been shown in my lab that the HD fetuses has an abnormal cortical development already at the 13th gestational week. And the overexpression of mutant Huntington in wild-type fetuses, embryos, was enough to recapitulate in adult stages the Huntington main features. So in those years, there were many studies that start to get interested in the development in HD individuals, and start to build together the hypothesis that an abnormal development could contribute to the adult disease. So one of the main structures that is altered in pre-manifestal HD individuals is the corpus callosum. That is the structure that is connecting the two cortical hemispheres. Has been shown that the corpus callosum has alteration at the level of the white matter tract, is having also abnormal network connectivity, and also the tractography is impaired. In 2015, Gatto and collaborators showed that that in the corpus callosum there was a decrease of axonal fiber, and this was not due to a neuronal death. So we hypothesized that the alteration of the corpus callosum in pre-manifestant patients could be due to an altered development. So the main technique we use to study specifically the callosal projection neuron is the neutral electroporation technique. This allowed us to target specifically this subpopulation of neuron with a reporter gene that can be the GFP, and after injecting the GFP in the callosal fiber, we are able to follow the morphology and the growth of the axon. The callosal projection neurons are mainly coming from the superficial layer of the cortex, and these neurons are generated at E15.5. So we inject the GFP with the electroporation at E15.5, and then we follow the morphology starting at P0, so immediately after the birth of the pups, until the end of their development. That is called P21, when the axons start to reach the contralateral side and start to establish connectivity with, to establish their own connectivity. So the first observation we had, it was that indeed in HD brain, the axons were shorter compared to the control one. And this decrease of axonal length was kept also at later stages. So also at P4, the axons were not able to recover the length and were still keeping being shorter. So the next question was why, which could be the consequences of this impaired axonal growth in adult stages. And for this, we analyzed the axonal density inside the corpus callosum, and we could see that the density was decreased, so means that less axonal fiber were crossing the corpus callosum. And at the level of contralateral side, we saw that indeed, even if a subpopulation neuron was able to reach the contralateral side, the connectivity was overall impaired to the control brain. So there were less connection point with the contralateral side. So indeed, we saw there was an axonal growth defect, and this axonal growth defect was having an impact in adult stages at the level of connectivity. So the question was, okay, why? Which could be the structure and the causes of this defect? We went to analyze a single-cell morphology, and we focus in a subcellular structure that is the corpus callosum, the growth cone, sorry. The growth cone is localized at the tips of the axon and is highly motile and is really in charge of pulling the axon and driving and guiding the axon to reach the proper target. As a special characteristic, the growth cone is at the level of the cytoskeleton organization. In fact, we have two main domains. The central domain that is mainly formed by bundle of microtubule and is really the core of the growth, is where the forces, the pulling forces are generated. And then we have the peripheral domain that instead is mainly formed by bundle of actin and is really sensible to the extracellular matrix. So as a role more in the guidance. Meanwhile, the central domain is really in the pulling of the axonal growth. So what we saw is that here are labeled the microtubule, and what we saw is that the growth cone of HD neuron were completely, the microtubule in the growth cone of HD neuron were completely disorganized, were not able to form a bundle, and also at the level of the peripheral domain, the exploratory microtubule that are the single microtubule invading and conducting the peripheral domain were really longer and not organized as in the control. So there was indeed a defect of the microtubule cytoskeleton per se. So to investigate which could be the molecules involved in this mechanism, we performed a mass spectrometry analysis doing gross cone fractionation starting from the cortices at P0. We obtain different proteins that were modulated. In green, there are the down-regulating protein, and blue, the up-regulating. And among the pool of the proteins that were down-regulated, there was NUMA1. That is a microtubule binding protein, really well known during cell division, and because it's organizing the microtubule cytoskeleton during the cell division, and is a well-known partner of Huntington. But the role and the function of NUMA in post-mitotic cell was completely not known. So the first step was to characterize the profile of expression of NUMA in wild-type neurons. And we stain NUMA, that here is represented in gray, together with the cytoskeleton. In magenta, there are microtubule, and in green, the actin. So what we saw that even also in a post-mitotic cell like the neuron, NUMA was strongly associating with the microtubule. So was enriched in the central domain, but was also decorating the exploratory microtubule, invading the peripheral domain. So NUMA, like during cell division, even in the gross cone, is associated with the microtubule and perhaps was participating to the organization. So since in HD, NUMA is downregulated, and also since its function is not known, we decided to downregulate NUMA in wild-type brain and analyze the different phenotype. Was really striking that with the SH NUMA, we could recapitulate all the HD phenotype we observe at the level of axonal growth and the gross cone. In fact, after NUMA, SH, the fiber were shorter, and the microtubule in the gross cone were strongly disorganized. So this means that NUMA is an important partner in the axonal growth, and the knockout of NUMA has strong consequences at the level of axonal growth. So now we saw that in HD there is axonal growth defect, that this axonal growth defect is due to this regulation and disassembly of the cytoskeleton due to a NUMA1 downregulation. But our goal was to try to recover the phenotype. So to restore the axonal growth and to recover in general, at least in part, the phenotype we observed, we were having two strategies. One was to overexpress the full-length NUMA. So acting really upstream to the pathway. But due to the size of NUMA, that is 250 kilodalton, we were not able to overexpress sufficiently the protein by viral injection. So we decided to go through another strategy that was acting directly downstream at the pathway, so at the level of the cytoskeleton. So we were needing to reestablish the microtubule array inside the cell. And for this, we screen several microtubule stabilizing agents, but we, let's say, then focus on the epithelium B that is non-chemotherapeutic, and we choose to work with the epithelium B because it's able to pass the brain barrier hematoencephalic, the brain barrier. And in addition, very few doses of epithelium B were enough to also promote axonal regeneration after spinal cord injury. So we inject our mice at P0 with a systemic injection of epithelium B, and then we sacrifice the mice 4 days after the treatment. And it was really striking that after 4 days of exposure to the drug, the axon were starting to grow longer compared to the no-treated brain, and were starting to recover the axonal lens. In addition, the treatment with the epithelium B was reaching the cytoskeleton and was able to rebundle the microtubule inside the gross cone of the axon. So with this study, we really highlight how in Huntington disease, during development, there is an alteration of the cytoskeleton per se, and this alteration in the organization of the cytoskeleton has a consequences in more adult stage at the level of callosal fiber. For the future, it will be really interesting to start to test drugs that are acting directly on the cytoskeleton to recover the organization, but also to test if the Huntington-related function with the microtubule could also be restored after the epithelium treatment. So I would like to thank my lab where I did my postdoc. It is in Sandrine Humbert lab, and particularly I would like to thank Rémi Carpentier, Radhia Kacher, and Eric Denarier that directly work with me in this project. My collaborator Marina Mappelli at the European Institute of Oncology, that is an expert of NUMA, the platform of proteomic at the CEA, and the laboratory of Frédéric Sodou for the all useful discussion during my postdoc. And thank you for the attention.
Thank you, Mariacristina. Is there any questions? Yes. Alors, I can run. There are fixed microphones.
Thank you very much. Microtubules are in lots of other cells, and notably in oligodendrocytes. So do you think you're going to see a difference in oligodendrocytes and myelination if you change your microtubules? And do you see that anyway, bearing in mind corpus callosum is highly myelinated?
I didn't check the myelinization of the fiber, but yes, for sure, when work with the cytoskeleton is always tricky in a way because it's changing a lot of, it's targeting all the cells, so we'll change the dynamicity of all the cells. But perhaps there also in the oligodendrocyte of HD there are defects. So in fact, it's never been explored this field until now. Yeah. So for sure will be interesting to analyze at the cell level of cell division already during development, and also in other cell type if there are defects, and if those treatment could help the myelinization of the fiber. For sure, yeah, with MRI will be. Because
we know that
really
the
nice.
HD mice have myelin defects, so it might
yes.
be interesting to have a look at.
Yeah, but it's also true that when we think to this wild phenotype, there are a lot of phenotypes, so a lot of defects. Perhaps the cytoskeleton is also acting in several of the points. So it's really this what I would like to do is to see if all acting on the cytoskeleton that is perturbed for sure, we can recover multiple phenotypes gathered from this treatment, in fact.
I agree. Be good.
Mariacristina, congratulations for a very nice presentation. Thank you. I'm perfectly in line with what you said about axonal and white matter issues related to Huntington disease. I was curious to know what you think about what do you expect to translate in terms of white matter in adult HD? We have been describing some kind of data you showed, Phillips et al. And the corpus callosum involvement. It's part of our work, and I perfectly agree with you. However, I'm not sure that the white matter reduced volume and the quality of white matter is so visible in the adult Huntington disease. Another question that I was just anticipated by the question before me, it was what do you expect to observe in terms of oligodendrocytes
I don't know
in the real human disease.
At the level of oligodendrocyte, I don't know. We didn't check, so we could check the cytoskeleton already in the cell, and it's true. No, but for sure it will be interesting to check the level of myelinization, but it's true that when you look at the tractography of the corpus callosum, inside there are also fiber. There is not only, so the density of that is slightly decreased, perhaps can be due to less fiber, and perhaps for compensation you will have an over-myelinization, so you lose the phenotype. Perhaps is an hypothesis. But I don't know. I have to check.
Look, one last question.
Congratulations on lovely work. Any evidence of peripheral nerve growth deficiencies in Huntington's?
No, we don't know. For the nerves, no. We didn't check.
Okay, thank you, Mariacristina.
Thank you.
Move to the next speaker.